Hello, everybody. Thank you for joining us, and welcome to the first webinar in AI in Pharma series by RSIP Vision. My name is Miki Chaimovic. I'm VP Business Development with RSIP Vision. And in this webinar, I will be hosting Moshe Zafran, our VP R&D. And we will discuss deep learning for the segmentation, classification, and quantification of dendritic cells. Some of us are still joining, but we would like to be on time, so we will start. A few things about RSIP Vision. We are providing AI and deep learning solutions for image analysis. The solution is customized based on your project needs and your data set. In that, we are different than many other providers in the market. We do not have some kind of a concept that was productized and now we are trying to sell it everywhere, but rather we take the opposite approach. We start with the customer and the customer's needs. We understand the problem or the question at hand. We review the data set, and then we customize a tailor-made solution for the customer. We've been doing that for a long time, over 25 years in the field with multiple repeat clients. Many of them U.S. corporations in the field of medical device, pharmaceuticals, and so on, as well as leading hospitals. So we have extensive experience in all AI and deep learning techniques in numerous medical and pharma applications. We have an experienced team, over 45 engineers in Tel Aviv, Silicon Valley, and Boston, as well as a medical team in staff to guide solution development and work with the customers. Partnering with RSIP Vision is relatively easy. It starts when you define what parameters and deliverables are needed from the solution. Then we build a custom solution based on the image data provided. Then the custom solution gets deployed for in-house use on your existing infrastructure. This is often a key parameter for working in the medical and pharmaceutical market, because information security and anonymization are very much a big deal in this market. And custom GUI can be deployed as well. I'm not going to tell you about all the things we've done. I will just say that many of them will find their way this way or the other to other webinars in our series. Just a few topics that we're thinking about are using AI in pathology, using AI in clinical trials, implementing AI in drug development processes, how to implement AI in your pharma company. I think that's a major thing for people who are AI fans. The fact that you are doesn't mean it makes things easier to do the buy-in within your organization, and we would like to help you overcome this obstacle. And of course, AI for curing cancer. A lot of very interesting things are happening in immuno-oncology, and AI could be impactful in those areas as well. As I said, we're working with leading hospitals, and one of them is Tufts Medical Center in Boston. That was a hospital with which we cooperated in order to do the project that we will be presenting now. Regarding convolutional neural networks for the classification and segmentation of in vivo confocal microscopy images. And I will hand it to Moshe to discuss this case with you. Moshe, the floor is yours.
Hi everybody, my name is Moshe. I'm the head of R&D at RSIP Vision, and I'll tell you a bit about this collaboration that we did with Tufts Medical Center on classification and segmentation of dendritic cells in confocal microscopy. So just a few words about the clinical background in the next slide. So dry eye disease is a very common medical condition. And so yeah, in the image we see now, we see a common diagnostic method with this dye that tends to adhere to the dry areas of the cornea. However, what people are looking for is a more quantitative way to measure and quantify this condition. And one way is to use confocal microscopy to look at the layers of the cornea, and specifically to look at a specific layer and to look for dendritic cells, which are immune cells that play a critical role in the pathogenesis and are a very good biomarker to evaluate the inflammation on the surface of the cornea. This diagram, we can see sort of a schematic of the layers of the cornea. So we have the outer layer called the epithelium, under that the Bowman's membrane and the subbasal neural plexus. The dendritic cells that we're looking for live only in this thin purple colored in the diagram layer. Below that we have the stroma, which actually comprises most of the corneal thickness, and underneath that an additional basal layer. The first step of this diagnostic process, when the doctor is using the confocal microscopy to look at the cornea, the first step is to understand if we're looking at the correct layer, because most of the thickness is not relevant to what we're looking for. So the user is going to focus the microscope at various steps, and we want an automated way to let the user know when he's looking at the subbasal neural plexus. After we do that, we can start to measure and quantify the dendritic cell level in order to get some quantification of the condition that we're looking for. So in this slide, we can see how typical images for corneal layers look like when viewed in this modality. So image column A is the epithelium. It looks like this. The subbasal plexus, which is the one we're looking for, looks like this. So there are nerve fibers, which are these long structures, and there are also dendritic cells, which later we will see more clearly. The third layer is the stroma, and layer 4 is the endothelium. So right away, even just intuitively, we can see that the differences in texture and in appearance between these layers are quite salient. What we did is we used a neural network architecture, a pretty standard architecture called VGG16. We add onto that various best practices and tricks of the trade that we learned were useful for these medical data sets, which are not as big as the data sets that these networks are typically trained on. And by training this network on altogether about 2,000 images in all of these layers, we can achieve very good classification accuracy in terms of sensitivity, specificity, area under the curve. These numbers in the table we just viewed before are all from a validation set. So we can get near perfect level identification of the different levels. And we hope that this will prove very useful in the first step of the process. So now we've identified automatically the subbasal neural plexus. So the image looks something like the image on the left. And our goal is to detect the dendritic cells. So there are these sort of 3 or more lobed linear structures that look like this. The long lines are not dendritic cells, even though superficially they may look similar. We want to detect them and measure them and do some quantification and reach the end result, something like we see on the right-hand panel. So how do we do this? So we also for this problem we use the supervised learning approach, neural network approach. Here we can see the basic architecture we used. Again, we modify it in various ways that we have found useful. This is a UNet architecture. So there's an encoder branch on the left in which the network is learning various filters and generating higher and higher level representations of the image, like conv, relu, pooling, rinse and repeat. And then afterwards, we have the decoder layer, which is learning how to upsample this and to generate the segmentation image. And of course, in gray, we have the skip connections that are letting the network use the low-level filter information for its final segmentation output. For this, we had a much smaller label data set because it's somewhat labor-intensive to label all these segmentations of the cells. So we use only 258 labeled images. We train the neural network on segmentation, most of them, and test on about 10% of the data set. And because it's a very relatively small data set, we do cross-validation. For segmentation problems, so we find that we can get very nice results, even using much fewer images, because in segmentation, each image patch or each image pixel is actually, we can think of it as an example. So instead of 2,000 images, here to get a nice quality result, a very useful result. It was enough for the segmentation task to use on the order of a few hundred of images. Okay. After we perform the segmentation and we know where the cells are, it's pretty simple to perform various measurements. So we can measure the density of cells, the number of cells per any unit of area. We can measure the area of each cell by looking at each pixel and doing some interpolation process along the edges to get a more accurate estimation of the area. We can measure the perimeter of the cell. Afterwards, so these parameters are actually what you want to calculate to quantify disease, right? So to quantify disease, the relevant measure is not the number of pixels that we have that agree with the ground truth. Of course, it's important to measure this, and this is what we're using to train the network. But in the end, we want to get, excuse me, a good correlation of these measures with our medical expert who guided the annotation of the images. So we let trained professionals label the image in a semi-automated manner using an image tool called Image J, and they can play with various thresholds, so they don't have to click on each and every pixel, and they can just correct the Image J results to manually annotate the data, and then we can do the same quantification and compare that to our fully automated result using our neural network. So when we measure our results, our quantifications of the area, the density, and the perimeter of the cells, and we measure the correlation between these values and the values from the expert annotation, again, we can get very, very high correlations. So in diseased corneas, all of these measures will probably be typically higher, and in healthy corneas, they will be lower. And getting a strong correlation indicates that we can use our automated method to basically reproduce what an expert would be doing with these images to do the quantification. Of course, this can be done very quickly. As the measurement is being performed, you don't need an expert to sit afterwards in front of a computer and spend many minutes or even hours annotating it. So it's we think this method has great potential as both fast and a reliable way to perform these measurements. Okay, so in summary and conclusion, so basically our goal is to help our clients. In this case, the client, so to speak, is the researchers from Tufts who we collaborated with. So what they care about is laser in vivo confocal microscopy a beneficial diagnostic tool that will enable non-invasive visualization of corneal layers and quantification of dry eye disease, and or at least of the dendritic cells, which are a good marker of this inflammation. And we're very happy with the result, and our collaborators are also very happy with it, and we're continuing to work together on further collaborations. We're very excited about using deep learning for this task, and of course for many other tasks to help the medical people and to help our clients perform automated diagnosis and really contribute to the medical imaging space.
Excellent. Thank you very much, Moshe, for this talk. We've reached the Q&A session. Some of you have already sent us questions. Let me just review those. Those who haven't sent questions and do have questions, please do send them. We'll be happy to answer as many of them as we can, as long as we're in time. And let me see here. First question comes from Jeremy. Jeremy would like to know, how generic are these solutions? Can this deep learning solution fit other needs? Moshe, what do you think?
Yeah, thank you, Jeremy. It's a great question. So, yeah, the holy grail, of AI and deep learning that everybody's dreaming about is to, get some general purpose AI, computerized pathologist or radiologist or ophthalmologist or whatever that can perform a wide variety of tasks. In practice, the technology is obviously not there yet. It's not a general purpose AI. Most of the basic solutions for these tasks are typically obviously task-specific, segmentation, classification, but also modality-specific. You train the network on a specific modality, and it's learning the statistics of these type of images, and that's where it's going to work afterwards. However, many people in the field, and ourselves included, are showing some nice advances in sort of, I would say, semi-general purpose AI. What I mean with that, it's still going to typically be task-specific. You're still doing segmentation task or a classification task or whatever it is, but there are ways to train these architectures to generalize somewhat across modalities. So there are very well-known challenges going on of, cell segmentation in a wide variety of staining methods, and each modality has different colors, different typical densities of the cells. Sometimes they're stuck to each other, sometimes they're not. The task is the same, but there are ways to train these networks to generalize across modalities. And once you've shown it enough of these modalities, then we can get the ability to generalize to a completely new modality that the network has never seen before. And of course, there are various data augmentation methods that can be used here to artificially even generate, new images that will help the network to generalize.
Excellent. Thank you very much, Moshe. The second question comes from Laura, and Laura would like to know, how long does it take to develop such a solution?
Yeah, that's a great question. So the simple answer is that it really depends. It depends on the data. It depends on how challenging the task is. And as a first step, we always sit together with our customers and do a deep dive into their data, understand both quantitatively and qualitatively how hard the task is, and then come up with some scope. What we do always try to do is to, once we understand the problem, is to define the scope in a way that some meaningful result can be seen, I'd say, within a month, to see that it's going somewhere, and some useful result within a small number of months. Of course, what useful result means and where that is relative to the full functionality that the client is looking for really very much depends on the project. There can be projects that after 2 or 3 months, we're done and we're very happy. And we have other projects for other clients that are, multi-year endeavors. It all depends on the situation.
Thank you, Moshe. The next question is from Michael. Michael would like to know, can we skip the training part? Can the deep learning solution teach itself?
Okay, yeah. So I would say the most mature branch of deep learning and AI these days is what's called supervised learning. So in supervised learning, the network needs a training phase. It needs some sort of labeled data set. However, this does not mean that we always need to work very hard to create the labeled data set manually. So as you can imagine, to label each and every pixel of these dendritic cells is very labor intensive and sometimes even impossible. Pharma companies can have basically unlimited data, but in many cases, annotated data for the training is quite scarce. And for this, we have various interesting solutions. So there's what's called, the bootstrapping method. So sometimes we will first develop a classical non-AI based algorithm that gives some approximated solution, or another way to think of it is automatic annotation of a training data set. So we can quickly develop some simple algorithm that will do it automatically, and we'll get some noisy result. Then we can train a neural network on that. And many times the network, after being trained on this noisy data, actually is better than the training data itself because it's generalizing across the noise. We can manually filter the noisy training data. So instead of the user or the annotator or the medical professional having to do all of the annotation work, we only need to throw out the mistakes or to do some correction. And then you can say rinse and repeat. So we have some neural network or some AI that's doing a decent job. We can look at its results. Again, have a human in the loop to filter out the problems and make some corrections or correct the accuracy in special cases that need to be retrained. And then we can retrain and repeat it. So to summarize, usually there are unsupervised architectures and this technology can be used, but it's, I would say, in general less mature than the supervised learning method, but even for supervised learning, the network's not going to train itself by magic, but there are ways to very efficiently create the training data without the customer having to work very hard in order to do it.
Thank you, Moshe. Next question is, what annotations or labels are required for the training images? Wen Liang would like to know.
Thank you. Yeah, so somewhat related to my previous answer. So the annotation or the labels basically are defining what output we want to see. So if we want to know what label of the cornea we're looking at, then each image requires, only one label. Which layer is it? Layer 1, 2, 3, or 4, and that's it. If we want to perform segmentation, then the network is going to require the segmentation to be labeled. So a mask of, where the cell nuclei are, where the background is, where the membranes are, etc. And then this data is used to train it on the task. If we're doing, what I called before semi-automated annotation, then you won't need to label it yourself. You'll need to talk to us about it. We will understand, how what the output should look like, and then we can figure out how to do it ourselves in-house or with some collaboration with the customer and or with our in-house medical professionals.
Okay, and the next question is from Pradeep, who would like to know how much data was required to achieve the 0.98 accuracy for this particular project.
Thank you, Pradeep. Yes, so we had two tasks here. The first one was to classify which layer we're in, and for that we needed in all about 2,000 images. So it means it was fairly balanced data set, so about 500 images per class. And in many cases, that's a good estimate of a typical minimal training set for a classification task. And in this case, the results, the accuracy was very, very high. Differences are salient, etc. On the other hand, for the segmentation task, we only required about 200 images in all. So a tenth of the number of images that were required for classification. And the reason is that, again, segmentation is looking at each and every area in the image, and there's an awful lot of information in a single image. So many times for segmentation tasks, much less data is needed to start seeing nice results.
Excellent. Thank you very much. And another question from Wen Liang. Can this method be applied to multispectral images with 8 bands? Not sure we can answer that. Moshe, yes?
Yeah, okay. So, of course, for multispectral images, many times it can make sense to do some pre-processing, unmixing, etc. But as an algorithms guy, so to me, it doesn't look all that different. So if you have one band or more that are reacting more strongly to what it is you're looking for, the nuclei or whatever, then one can apply AI to that single band. If all the bands contain useful information, then technically it's very easy to use a multi-channel image as the input to the network and to train on that. So yes, absolutely. These methods can be applied generally to multi-channel and multispectral images.
Okay, thank you. A question from Yoram. Can you find trends with your deep learning?
Can we find trends, Vicky?
Yep, that's the question. Maybe, Yoram, if you can clarify the question, it will be easier for us to answer. We can move to the next question. Do you have to crop all the single dendritic cells as inputs for the neural network?
Okay, so in terms of the input training data, so again, the training data is represented as a binary mask. So each pixel is either belongs to a dendritic cell or does not belong to a dendritic cell. Either it's just an object or a background. And this was labeled again in a semi-automated manner. So the annotator didn't have to manually separate the cells from each other. They just had to play with various sliders and some image analysis old school program to create this result. So if two cells are not touching each other in the binary mask, then obviously they'll be separated from each other. In this case, there were not too many problems of cells that are touching that needed to be separated. In other cases, of course, that's a very important part of the task. And in cases such as that, when cells are touching each other or nuclei or whatever it is, then we can either do instant segmentation, which is different architecture than the one I spoke about, or we can do various tricks for units. So instead of training the network just to learn every pixel of the segmentation, we can also train the network to learn what are the borders between the objects. And then if the network knows where the border is, then we also get the separation as an automatic byproduct of the segmentation.
Excellent. Thank you very much, Moshe. Last question comes from Yuan. What makes RSIP special? Why are you better than your competitors? I'll take this one myself. Firstly, what do you mean? Everything about us is special. But seriously, if I have to say, I'll mention two things, that we customize the solution that you need, and that we have a large experienced team led by Moshe. That would be a good opportunity to thank you all, to encourage you to follow us on LinkedIn and find out about our upcoming webinars, and to contact us if you have any other question whatsoever. Thank you very much, people. Bye-bye.
Thanks, everyone. Pleasure.